Good morning and welcome to today's broadcast of the MPG Primer, live from the Broad Institute in Cambridge. This morning's primer will begin at approximately 8:30 Eastern Standard Time. Until then, we may be doing some audio and video testing to make sure that your live stream is working to the best of its abilities. Please stay tuned. Again, our program today will begin at approximately 8:30 a.m. Thank you. I completely blanked on the fact that you guys were in here today.
Isn't it great?
Yeah, it is. I wish you guys were doing it in here every day.
I know. Hey, you know what I'm gonna do is so I asked late last night as my program organizer at the Eleanor, we're still putting the slides together.
Yeah.
We decided we were going to put. They had 3 people speaking at the second meeting.
Yep.
And everyone with the phone. So I think now I'm going to just try to get myself comfortable with toggling from my laptop to the phone.
Okay.
Just because I'm probably going to have to stay here just to make sure.
Okay.
Because I don't know at what point they're going to get onto the app.
Sure.
It needs to be like super.
Yeah. Well, and
I know it's just like switching the plugs out,
right.
but I don't. Is there some? Do you think like we'll have to make an adjustment on hit left turn, left turn, left turn, left?
All you would essentially do is, so if you have,
I'm gonna grab mine.
yeah, did you happen to grab that adapter? You did, okay.
Actually, it's on the top shelf under the podium.
Oh, okay. And actually, it could probably stay down here. It doesn't have to be up there for any reason.
Okay, good morning, everybody. Can you all hear me?
Yes.
Yes? All right, fantastic. My name is Neena Pyzocha. I am a graduate student in Zhang Lab, and today I'm excited to give a talk about genome editing tools and applications. So here is an outline of what I plan on discussing today. First, I'll give a brief introduction to genome editing and CRISPR-Cas9 tools. And then secondly, I'd like to provide some practical tips for using CRISPR nucleases in your lab and then show an example of these tips from the literature. And then lastly, CRISPR-Cas9 has a suite of very useful tools, and I'd like to highlight some of those in the third part of the talk and go through some of these applications that may be of interest to you and to your lab. And so to start with an introduction to genome editing. First, how is it possible to even edit a genome? To do genome editing, we take advantage of processes that are naturally present in mammalian cells to repair double-strand breaks. And these processes are there because it's very important to be able to repair double-strand breaks because this can be very bad for a cell to lose part of its chromosome. And so there are two main pathways for repairing a double-strand break. The predominant pathway is NHEJ, which stands for non-homologous end joining. And that is a process in which a double-strand break is ligated back together. And however, it's often a very error-prone process and can result in insertion or deletion mutations or indels. If this type of double-strand break can occur within a gene, an indel mutation, if resulting in a frameshift mutation, will often lead to a premature stop codon and therefore knockout of a gene. And so this is how you can knock out a gene using double-strand breaks. The second pathway is HDR, or homology-directed repair. And this type of repair requires a template, but results in precise gene editing. HDR is very nice, however, it doesn't happen in all types of cells. For example, adult neurons, which do not replicate, essentially do not do HDR. And so now that we know that we take advantage of these double-stranded breaks to do genome editing, how do we create artificial DNA double-strand breaks? And the way that we do this is using programmable DNA nucleases. Programmable DNA nucleases aren't an entirely new thing. In the past, people have used zinc fingers or talons, and these work quite well. However, their DNA binding is provided by modular protein domains. And so to create a specific zinc finger or talon would require laborious molecular biology processes to clone together multiple little fragments. And so this was a long process, and you had to engineer every individual protein for every target site. CRISPR, on the other hand, is an RNA-guided DNA cleavage, and it consists of a protein and a targeting RNA. Therefore, if you want to target different genes, all you need to do is to change the targeting RNA, which is relatively simple and very quick. So CRISPR was harnessed from bacteria, and it's a naturally occurring bacterial immune system. CRISPR is composed of two main components, the Cas genes, which are proteins, and then the CRISPR locus, which is alternating repeat spacer units. These repeats, when transcribed, form these hairpin-like structures that are recognized by the Cas genes, and the spacers are specific to the particular phage that they can target. And so let's say a bacteriophage infects this bacteria. In some instances, a piece of the phage DNA will be integrated into the CRISPR locus. And then if infected again, or after in the locus, the CRISPR locus is transcribed into this long pre-CRRNA and then processed into individual units, which then associate with these Cas genes and target its specific virus. And so I just described the basic way that CRISPR is used as an adaptive immune system in bacteria. And that was one example, and there are many different types of CRISPR systems out there. But they are placed in two main categories, which are important for genome editing. And so the first is class 1. And the main distinction between these two classes is that class 1 is composed of a multi-subunit effector complex. So you need multiple proteins to come together to associate with the RNAs for targeting. And then you have class 2, which is just a single but large protein. And so for genome editing purposes, we always use class 2, or these large single effectors, because it just makes it simpler, having a two-component system versus a multi-component system. Cas9 was the first and is also one of the most well-known enzymes for CRISPR-Cas, and it is an RNA-guided DNA endonuclease. Its crystal structure is shown right here. It's a bilobe structure, and it has two main nuclease domains, the RUV-C as well as H and H. Each one of these individual nuclease domains cleaves one strand of the double-stranded DNA. The first and most well-known Cas9 comes from the bacteria species Streptococcus pyogenes, and therefore Cas9 is also, this particular Cas9 is also referred to as SP-Cas9. And again, it's a two-component system consisting of the protein and an engineered single guide RNA. So here's a schematic of using SP-Cas9 for genome editing. And there's a few things I'd like to point out. So first, again, you have SP-Cas9, which is the main enzyme involved in cleaving the double-stranded DNA. And it's targeted using its guide RNA, which has this complementary sequence which Watson-Crick base pairs with its target. And then the guide RNA scaffold, which is this hairpin-like structure that the protein recognizes to bind and associate with the RNA. Another key component for genome engineering is what we call the PAM, which stands for the protospacer adjacent motif. And it's a sequence that's required for Cas9 binding and subsequent cleavage. And so one important reason why the PAM exists is that if you, I can go back to this slide, the CRRNA is encoded in the bacteria's own genome. However, you don't want the bacteria to cut its own genome because that could result in death and would be very, not very good. And so the PAM is one way, or it's an important way so that the bacteria can identify a viral phage genome versus its own. And the PAM is required for Cas9 binding. So to further go through these and talk about kind of the mechanism of DNA binding and cleavage, here's a nice schematic that I like to refer to. And it's also important to go through this because it relates to the specificity of Cas9 genome editing, which is very important. So again, you have the enzyme complex with your CRRNA, and then you have the double-stranded DNA. The first thing that the enzyme with its guide does is that it recognizes the PAM and binds to it. And then it starts to undergo DNA melting proximal to the PAM and then outward. If there is extensive pairing, DNA cleavage occurs. If there is just partial pairing or there's multiple mismatches, there's no DNA cleavage. And so again, this directly relates to specificity. And so since the PAM is required for binding of Cas9, if there is a mismatch in the PAM, then Cas9 won't bind. So this is very critical. Also, the bases directly next to the PAM are very important. And then Cas9 also does not tolerate multiple mismatches or indels. However, if there are mismatches towards the distal end of the PAM, these are more likely to be tolerated and could still result in cleavage. And then lastly, if there's a single base mismatch or indel, this could also possibly result in cleavage. So any questions before I move on? Okay, so here are some practical tips for using CRISPR nucleases in your lab, and then I'll go through an example, kind of like walking through these practical tips. And so when considering using CRISPR in your lab, here is a checklist that I like to go through when doing a targeted genome editing project. First, it's very important to consider your project's needs. Is off-target cutting very detrimental to your final product? Are you planning on using a very specific delivery method? And then lastly, do you have sequence constraints that may suggest that you use certain types of CRISPR nucleases? And so once you have an idea of some of the needs of your project, you can start to explore which CRISPR nuclease to use. And then after you choose your CRISPR nuclease, because that will change which PAM you are working with and targeting, then you can use computational tools to identify your targeting sequence. And then lastly, once you've chosen your guide, it's very important to test for on-target and off-target DNA cleavage. There are many tools that are available to the research community. Some by our lab, the Zhang Lab, and then many other labs too have deposited their genes to add genes. So I highly recommend, if you're interested in using CRISPR in your lab, take a look at AdGene. There's tons of different plasmids. They can target multiple different species. They have many different things that you can do with them. But for the sake of right now, I'm just going to focus on if you're looking to do targeted genome editing. And so you're looking to cause a double-strand break and cut the DNA. And so in many cases, enhanced specificity or having a strong specificity, so low off-target activity, is critical for your experiment. And there are two main methods that you can use to have the highest specificity in your genome editing experiment. And so the first is that you can use a 2-Cas9 enzyme system. First, we have paired Cas9 nicases. And so if you remember, Cas9 has these 2 DNA cleavage domains, H and H and RUBC. And if you inactivate one of the domains, it will nick a single strand. You can pair these nicking Cas9s and put them in close proximity and then nick opposite strands, which results in a double strand break ultimately. But if you have a single off-target nick, that is less likely to cause any damage. And so this results in higher specificity. Another one developed utilizes dCas9 or dead Cas9. Yes, question.
Do both Cas9 recognize a single PAM, or there have to be 2 PAMs?
Oh, I see. Yeah, so that's a good question. So the question was, do the Cas9s need to recognize the same PAM, or do they need to recognize different PAMs? So I believe the one that we used recognized the same PAM, but it would just target 2 different sequences. You can. This is a bit of an aside, but you can use multiple different Cas9 or Cas9 orthologs or CRISPR nucleases to do to target different regions in the cell to have and do different processes. So you can do multiple different Cas9s. Yes.
And so your diagram before sort of indicated that it binded to Cas9, and then the other end of it separated the strands.
Mm-hmm. And so the second one utilizes these dCas9s. So these are catalytically inactive, so they're just simply Cas9s that can bind to the DNA, but they're fused to these FOC1 nuclease domains, which upon dimerization can cleave the double-stranded DNA. These are very nice approaches and they work quite well. However, one key disadvantage is that you need 2 individual guides and they need to be within a close proximity to each other to result in efficient double-strand breaks and genome editing. And so if that is not of interest of you, you can also use these other enzymes, which are enhanced specificity single Cas9 enzymes that have undergone structure-guided protein engineering. And there's two different versions. One is called ESP-Cas9, and the other is called SP-Cas9HF1. And they have different mutations, but ultimately the amino acid mutations are they interact with the targeting regions, or close to the targeting regions of your guide RNA, and therefore these papers have illustrated that there is higher specificity. So you have clean clean on-target cutting and reduced or eliminated off-target. When referring to Cas9 in previous slides, I was always referring to SP-Cas9, again, the Cas9 derived from Streptococcus pyogenes. There are other Cas9s in other bacteria species, and one especially notable one comes from the bacteria species Staphylococcus aureus, and it's called SA-Cas9. The key difference here is that it is much smaller, and so it's just above 1,000 amino acids, whereas SP-Cas9 is above 1,300 amino acids. And although 300 amino acids may or may not make a huge difference, the reason why this makes such a big difference is you're able to package SA-Cas9 with its targeting guide and necessary promoters within the region needed to fit into an AAV vector. And so if you're working with AAV and that's your delivery method, SA-Cas9 is a very useful tool. Its PAM is a little bit different, so it's a little bit longer, and so that could be a little bit more difficult depending on how tight your sequence constraints are, but that's just one component. And then lastly, there is a completely new enzyme, so it's not related to Cas9 at all, and it's called CPF1. It's another CRISPR-associated single effector DNA nuclease. And it has RevC domains similar to Cas9, but again, completely different amino acid layout. It has a smaller CRRNA. And one other interesting thing to note is that it can process its own CRISPR array. And so here's a table with the key findings of the last few described Cas9s and CPF1s. And so S. pyogenes has a very short PAM. It's very well characterized, and so this is often one people use to do genome editing. But if you're interested in using AAV vectors, SA-Cas9 is very nice in that it's small and you can fit a single guide and the Cas9 within a single vector. The PAM is a little bit longer. CPF1, one key advantage is both of these PAMs are G-rich. And so if you're working in an area of the genome that is T-rich, CPF1 has 3 Ts for its PAM, and so in some cases, CPF1 may be better. It also results in a stagger cut, and it can cleave its own CRISPR array. And so that could be advantageous too. So once you select the Cas9 or CPF1 that you're working with, the next thing you need to do is to design your targeting guide sequences. And the best way to do this is to use any of the available online tools. And there are many of them, as shown from this nice review by Tycho et al. They all have differences, and and depending on the application that you're doing, may be more or less useful. Some have multiple species. Others consider off-targets that have indels, which is a new, relatively new finding. Our lab has designed the MIT CRISPR design tool, so that's the one I'm most familiar with. Another colleague of mine recommended Cas-Off Finder, but it might be good to review multiple of these and see which one best fits your needs. So here's an example using the MIT CRISPR design tool. And so the way that you do this is you select the region of DNA that you would like to undergo genome editing. If you're simply knocking out a gene, we highly recommend going with the first constitutively expressed exon within the gene. And so you input this DNA into the search algorithm, and it'll go through and calculate the probability of different off-targets and score them, and then show where it's located within your region of interest. And this is nice because it shows which off-targets, it predicts which off-targets may result. While these off-target predictions are based on what we know about Cas9 binding, sometimes it's empirical, and so it's important that you screen for these too. And one of the quickest ways and most informative ways to test for on-target and off-target cleavage is to just use NGS. And so with NGS, all you need to do is to design primers that flank the region of your targeted DNA editing, and then you can also look at some of your off-target sites too. Pool these together, and here is an example of some of the indels that you may see if you do targeted genome editing. And so with a cut site, as indicated by this red arrow, you can have small deletions, or you can have some small insertions. Also, you note that this varies cell to cell, so you can get multiple different indels. It's unlikely that you're going to have the exact same type of deletion in every cell. In some cases, it may be important for you to know the entire landscape of off-target cutting. And so to do this, one way that you can do this is through unbiased off-target detection methods. And there are several that have been reported in the literature. You can do anything from sequencing to specifically pulling down double-strand break regions, capturing inserted sequences that are put in during repair of a double-strand break. And a lot of these have their own pros and cons, but they all are effective ways to search for double-strand breaks in an unbiased whole genome fashion. A common thing to do with genome editing is to do HDR. And so I'll briefly go over how to do HDR using CRISPR-Cas9. So again, HDR is a method used to make precise genomic edits. For HDR, ideally, you want to have your CRISPR nuclease cut close to a target of interest. And then you need to provide a template for your specific mutation or insertion. And here's one key thing that I like to tell when discussing HDR. CRISPR generally works well in most cell types. However, HDR efficiency really varies depending on the cell type. For example, in mouse ES cells, it works very well. Whereas, again, in adult neurons that are not dividing, it essentially doesn't work. And so this is very cell type dependent, and so HDR can really be the rate limiting step in this process, not so much CRISPR. Again, you need to have an HDR template in order to do homologous recombination. And these homology templates will consist of your insertion or your mutation flanked by homology arms. And these homology templates can either be single-stranded oligonucleotides, for example, purchased through IDT, or you can use plasmids that are linearized. One key thing to note is that your cut site should be close to your HDR site. In this one example here, the authors found that approximately a 30 base pair window of your cut site to your modification site is optimal for HDR. So here's a brief walkthrough of designing an experiment to do HDR. And this is just a proof of concept one that was done in a Nature Protocols paper by a member of our lab. And so the goal here was to insert a Hindi3 restriction site using either a plasmid repair template with 1 kilobase homology arms, a single-stranded oligonucleotide repair template, either the sense or the antisense strand. And so these underwent HDR and cutting. This is the guide that we used, and you can see the cut site is right here, and here is the insert. So again, very close cut site to insert. And this insert is very small, and so we had these short arms for the oligonucleotide. And the results down here illustrate the HDR efficiency. In HEC293 cells, yes, question? Yes.
How do you deliver a single strand?
So this was just by transient transfection. So depending on your application, that may vary. Sorry, could you repeat that? How much template?
Double-stranded template be used instead of single-stranded?
I assume so, yeah. I didn't run this experiment, but it's possible that can be used. It might be good to read through this paper and see what they recommend too. So the results show that in HEC293 cells, so these are very well transfected cells, you can have up to 27% HR, which is actually pretty good. This is using the wild type Cas9. D10A, that means that we're using the nicases. So overall, nicase activity very much reduces HDR efficiency. And so you have a bit of a trade-off between cutting efficiency and targeted activity. In Hughes 9 cells, these are human ES cells, we were able to also get HR, but at a lower rate. So one final thing I'd like to note, here is the picture again, illustrating the two different pathways by which double-strand breaks can be repaired. And again, homologous NHEJ is the preferred way. But if you're trying to do HDR, one way that you can promote it is by blocking NHEJ pathways. And there are some that have been reported by the literature that you can suppress expression of NHEJ proteins, for example, by shRNA gene silencing. There's also this known ligase 4 inhibitor that can also be used. And so here's an example kind of putting this all together as an example for targeted gene editing. In this paper, the goal was to have in vivo targeting of PCSK9, which is a gene involved in cholesterol regulation, targeting the mouse liver. And so that's the project goal. And so since we're doing in vivo targeting, the authors decided to target with AAV8. And so therefore would require Staphylococcus aureus, SA-Cas9. AAV8 is also specific tropism to target the mouse liver. And so here's the design. They drove using a TBG promoter, which is again specific to, I believe, hepatocytes. You have SA-Cas9 and a single guide which fit within the amount of size required for expression and to be targeted using AAV. Here's their experimental workflow. First, they had in vitro target validation, so they made sure that their targets worked before going into the mouse. And then they produced virus, did tail vein injections, and then tissue analysis. And what they found is that 7 to 28 days post-injection, they had about 40% indel efficiency, and so they had quite high activity. And not only did they see indel activity, but this resulted in a phenotypic change in the level of serum cholesterol. So there was a significant drop in serum cholesterol in mice that were treated with SA-Cas9 targeting PCSK9. Again, an important thing to look at is off-target cleavage. And so the first thing they did was they used BLESS, which is one of the unbiased whole genome methods to detect for off-target cleavage. And they found that they were very lucky in that the particular guides that they chose were very specific. And these Manhattan plots showed that there was strong enrichment at the on-target locus alone, with very minimal to no other regions that were identified. And then after injecting the mice with virus, they harvested liver tissue and then did targeted NGS sequencing of this tissue at the on-target and predicted off-target sites from this BLESS assay. And they found targeted DNA cleavage and little to no off-target cleavage. So in this last part of the talk, I'd like to go over some other CRISPR applications besides just using Cas9 to edit genomes and to do HDR. First, one important thing I would like to discuss is that there has been a CRE-dependent ROSA26 Cas9 knock-in mouse. And so it has a LOXP stop site, and so you can control which tissues it's expressed in, and inducible by CRE recombinase. And the reason why this may be advantageous is that if you want to target multiple genes, you can just deliver your guides only. It's also important to note that these mice are viable, fertile, and they present no known morphological abnormalities. And you can restrict expression to a certain tissue, or you can express it across multiple different tissues. dCas9, so again, the Cas9 that has had both of its DNA cleavage domains inactivated to be a DNA binding protein. So dCas9 can also be coupled with transcriptional activators, leading to a programmable transcriptional activator process. So the way that this was done was that after the crystal structure for Cas9 came out, it was examined, and in a structure-guided way, a fusion of VP64, which can promote transcription, was fused to dCas9. In addition to that, the guide RNA was also analyzed and was found to have certain areas of the guide that were amenable to adding sequences. MS2 loops were added to these open areas, and MS2 stem loops can be strongly bound by this MS2 binding protein, which was fused to additional transcriptional activator domains, HSF1. These can be used to upregulate mRNAs, link RNAs, and have been found to be quite effective. Another very useful use of Cas9 is to use Cas9 screening methodologies. You can design a library, either a library of all the genes or specific genes of interest to you, clone your library into integrating lentiviral vectors at a low MOI, and select for cells that received a lentivirus, and then apply a selection pressure to screen for certain genes of interest. So for example, a few colleagues in my lab screened the A375 melanoma cell line, selected for cells that were resistant to vemurafenib, which is a therapeutic BRAF protein kinase inhibitor, and found new and previously reported candidate genes that were involved in resistance. And so knockout screens can be applied to many other applications. Cas9 screens can also be used with these previously described transcriptional activators. And so you can have an activation screen or you can have a knockout screen. Furthermore, there's been a very recent paper that came out in Nature Method have combined DropSeq, which has also been done here at the Broad, with CRISPR screening. So you can now do single-cell CRISPR screening. And so this is a new and powerful technique that may be of interest. And so I highlighted a few of the DNA targeting applications. And there are a few other ones that are worth mentioning that I didn't go fully into. You can also do some epigenetic modulation and affect histone modifications. You can label DNA. Again, screening is very powerful. You can also do transcriptional control. And lastly, before the talk concludes, here is something that's near and dear to my heart. But we have recently discovered that there are new RNA targeting CRISPR nucleases. The first one discovered and profiled was C2C2, also known as Cas13a, and the second one is Cas13b. Both of these enzymes have functional domains that are HepN domains, and they cleave single-stranded RNA. An interesting thing to note is that both of these enzymes also have this property called the collateral effect. And what the collateral effect is if you prime cleavage of these enzymes. And so if it has on-target cleavage, once it has that initial on-target cleavage activity, it then becomes this non-specific RNase, which is perhaps not ideal if you're trying to just knock down a certain RNA, but could be useful for other processes. And so the collateral effect could definitely be useful as a diagnostic. And so if you have a pool of RNA, you're trying to see if there's a certain RNA that's available or within your population, you could be able to screen for that, and that has been done in this paper here. Also, these proteins, if you knock out their hep-end domains, become very nice. You can target these enzymes to the RNA as targetable RNA binding proteins. And you can fuse multiple different effectors that could have interesting things. And so you can do potentially, you can repress translation, enhance translation, you can look at localization, and so on and so forth. I should note that these are in the works right now, so they're not currently available, but I just wanted to end the talk with kind of with the future and things that we're hoping to do with RNA targeting CRISPR tools. So hopefully throughout this talk, I was able to convince you or to provide you some additional knowledge how CRISPR enzymes can be very useful for multiple different areas of science. And we hope that these tools will continue to be widely useful to the scientific community. So here's my lab. They've done some really fantastic work, and it's been a great place to work. And with that, I can take any questions. Yeah?
So you mentioned in vivo and mice, with AAV, there's targeting of some liver cells.
Mm-hmm.
Is there a way to target, or at least to ensure that the efficiency is high enough of germ cells?
Of germ cells. So the question was, can you ensure or see if you can target germ cells? And so, again, Cas9 generally works well in most cell types, but I think the key thing here is delivery. And so if I unfortunately am not familiar with working with germ cells, but if you look through the literature, there may be certain experiments that are good for targeting to germ cells. And so as long as you have a method for effective delivery, then if Cas9 can get into these cells, it's likely that you can have targeted DNA cleavage. So it's more of a delivery question, unfortunately. Okay. Well, I'll be up here if you have any other questions. So.